My name is Caitlin O'Brien, and I'm a PhD student at the University of Caen in the Ethos Laboratory. Today I'm going to talk to you a little bit about the effects of prenatal stress on offspring behavior in the cuttlefish Sepia officinalis. Stress is the subject that's of great interest to behavioral biologists for a number of reasons. Loosely defined, it refers to the physiological response an animal has when it perceives a threat. And the reason that this is of such great interest to behavioral scientists is because of its long-reaching effects on animal survival and fitness. In many cases, the stress response actually improves survival. It allows the animal to combat whatever was causing the stress in the first place. But in many cases, it's also associated with negative side effects that detract from overall fitness. Within this broad topic, one subject that's developed a lot of interest lately is the subject of maternal stress. And this simply refers to stress that is applied to a pregnant or spawning female. There have been a number of studies in recent years looking at this, with mainly rodents and other mammals. And they've found a variety of effects on offspring behavior, including increased fearfulness, reduced exploratory activity, delayed motor development, and also impaired coping abilities. Now, this also leads to the question, does stress applied directly to developing embryos also have an effect? And unfortunately, this is somewhat difficult to test in a lot of species since the young develop within the mother. But the studies that have been conducted, mainly with reptiles and birds, have shown a variety of effects, including reduced activity and also greater shelter-seeking behavior. So, on to the cephalopods. The species that I study, the Sepia officinalis, the common cuttlefish, we can actually study the effects of both types of stress because the young develop outside of the mother. And more interestingly, work in our lab has actually shown that the late-stage embryos can perceive visual, chemical, and somatosensory stimuli in the environment, which gives us a broad range of stressors and stimuli that we can test. So in order to determine whether or not embryonic and maternal stress affect offspring behavior, we devised a two-phase test. In the first phase, we collected wild cuttlefish, split them into two groups. One group was set aside as a control, and the other group was subjected to handling stress on a daily basis. We then allowed them to lay eggs and collected those eggs and allowed them to hatch. For the second phase of the experiment, we took the eggs from the untreated females and split them into three groups. And the first group was a control group. The next group was raised in contact with a natural predator, the sea bass. And the third group was exposed to bouts of bright LED light on a random basis throughout the day. So after hatching, we allowed the eggs to hatch, and we conducted a battery of behavioral tests on the offspring. Our first test looked at body patterning. We placed 1- and 2-day-old cuttlefish on a uniform gray substrate and on a black and white checkerboard substrate, and we measured the overall disruptiveness of their body patterning and response. Now, based on work with adult and juvenile cephalopods or cuttlefish, we had actually expected that they would use a background matching strategy so that they would show a low disruptive score on the gray substrate and a high disruptive score on the checkerboard. But as you can see, there's very little difference within groups between the two background types. So it appears that actually instead these young cuttlefish, they're 1 and 2 days old, are actually using a different type of camouflage strategy, perhaps disruptiveness. We did, however, find that within the stress mother group, they did express a much higher level of disruptiveness on both backgrounds, which perhaps indicates a stronger defensive response. In our next test, we looked at predation ability in four-tailed cuttlefish. We put them in an arena with a small shrimp and filmed their interaction. We then measured the capture rate, so the proportion of each group that managed to capture a shrimp, and also the success rate, so the percentage of all of the attacks that were successful versus unsuccessful. So here we see the results from the second phase of the test with the, oh, by the way, I should mention that if you don't see a graph, it's because there was no significant difference. In the interest of time, I'm only showing the interesting results. But in this graph, we see the results from the second phase of the test, looking at the capture rate on the y-axis. And here you can see that where the control and the predator groups had between 30 and 40% capture rate, the light exposed group was nearly double that, and this was very significant. Likewise, when we looked at the success rate, we found that where the control and the predator groups had about an 80% success rate, the light exposed were actually near perfect. And what we believe is happening here is that the additional light that they were exposed to during development accelerated visual maturation and gave them stronger predatory abilities later on. For our next test, we looked at overall activity levels. So we placed cuttlefish in an open arena at midnight on their 9th day of life, and we filmed them for the next 6 hours. We then used the behavioral tracking program EthoVision to measure the total distance traveled as a proxy for overall activity. In this graph, you can see the results from the first phase of the test, looking at maternal stress. As you can see, there's a very big difference between the two groups. On the y-axis here, we have the total distance traveled in centimeters over the course of the 6 hours. And as you can see, the stressed mother offspring traveled nearly twice the distance over the course of that 6 hours. Now, this was a little unexpected because we would expect an animal that's more stressed, and particularly a cuttlefish, which relies on camouflage, we would expect it to remain more still relative to the control group. So possibly what's going on here is they're searching for shelter in the open arena or some other effect. Finally, we looked at the threat response in our 12-day-old offspring. We placed them again in an open arena, filmed their baseline activity for 1 hour, and then we added either blank seawater or predator odor and filmed the activity after that for 1 hour. We then divided the distance traveled after the treatment by the distance traveled before the treatment to standardize between individuals. What we found was on the y-axis here, remember it's distance traveled expressed as a percentage of the initial distance. And as you can see, there's a very strong reaction for the stressed mother offspring compared to the control mother offspring. They almost completely stop moving in response to the predator odor. And this is interesting, especially in light of the previous experiment, which showed that these same offspring tended to move more in a non-threatening situation. So obviously more exploration of this is needed. So to recap, maternal stress was associated with higher disruptiveness in the camouflage and the body patterning, had no effect on predation ability, increased activity in a non-threatening scenario, and also resulted in a stronger threat response. By contrast, the predator odor had no effect on any of these measures, which was particularly surprising since of the 3 stressors that we tested, we actually expected this one to be the most relevant and thus to have the strongest effect. But what we think might have happened here was the exposure during embryonic development was continuous, so it's possible that some form of habituation occurred to the predator odor. finally, the bright light, especially relevant in light of the last presentation, had no effect on body patterning, but it did dramatically improve predation ability, which again we think is a factor of visual development, and no effect on overall activity levels or the threat response. So, what are the implications of this? Well, for one thing, we now know that we can use Sepia officinalis as a behavioral model to study stress. This is important in my larger thesis project. We're looking at a variety of species, zebrafish, quail, chickens, in order to study the interplay between these two different types of stress, which you can't really study in mammals. And interestingly, for me at least, was the fact that the maternal stress seemed to have a greater effect on the behavior of the offspring than the embryonic stress. Before the experiment was conducted, I actually predicted that the direct stress, the embryonic stress, would be more relevant since it would be a more direct predictor of future environment. But instead, as we often are in science, we were surprised. So, a number of wonderful people to thank, especially my advisor, Dr. Ludwig Dickel, and some of the many wonderful people pictured here. Thank you very much.